Hello, welcome to 13 Universal Learning Beyond Barriers, and this is A-level Caie Biology with shared role. So we are in all topics, paper 4, series 6. Okay, sit tight, let's go on a journey. Please bring out your writing materials. Okay, let's apply some concept. The first question here, of course, this is paper 4, A2 Biology. So sarcomeres are functioning units of myofibrils of the muscle fiber of striated muscle. The arrival of an action potential at the sarcoplasmic reticulum of a striated muscle fiber can lead to the shortening of sarcomere. This shortening occurs by the sliding filament model. Outline the sequence of events occurring after stimulation of the sarcoplasmic reticulum that results in the shortening of a sarcomere. All right, so this is it. Okay, you can see, so these are the events that occur. All right. So the first thing that happens is that the calcium ion channel opens. Remember, so remember that the arrival, okay, so arrival of, you can see that, of action potential causes in the sarcoplasmic reticulum causes calcium ion channel to open, and the calcium ions leave sarcoplasmic reticulum and then binds to troponin. Okay, then troponin changes its shape. Okay, yeah, and then of course, and when it changes shape, it moves tropomyosin, exposing binding site on actin. So of course, once exposed, myosin head will bind to it, and then cross bridge is formed, myosin head tilt, and then we have stroke movement, guys. So this is it. So memorize it. Okay, this is a sequence of events that occurs. Let's go to another question quickly. Scientists compare the diameter of samples of striated muscle fibers taken from young mice and adult mice. So look at that. So you can see this is the results shown in fig 7.1. So this is number of muscle fibers. This is muscle fibers diameter. You can see the key. Now, before you start analyzing, of course, let's look at the question says, with reference to fig 7.1, describe two differences. Okay, two differences between the muscle fibers of young mice and adult mice. So just how these differences may affect sliding filament model. Okay, so two differences between the young and the adult. Okay, now guys, stop this video, pause the video, and try to solve the question. I'd like you to apply concept, try to solve this question. Okay, so this is the analytical skill that you're looking for. But before you go on, guys, always look out for the value of one box. Okay, look at this. From 10, now this is 10 to 30, that's about 20. Okay, from 0 to 20 is about 10 boxes. So for this axis, you see that 10 boxes, okay, gives us 20 units. Okay, you can work it out. One box will give us what? That will be 20 over 10. So one box is 2, right? So work out this. So if 10 boxes gives us 10 units, so one box will give us 1. So each box has 1, each box has 2. Okay, very important. So what do we do, guys? So what you do is, because it says describe two differences. Okay, so if you look at this, you see that for young mice, one of the differences here is that, look at the range of the diameter. The range of diameter here is from, this is 10, 12, 14, about 14 or 15. Okay, 15 to because each box is to about 46. All right, 15 to like 46. Okay, that's the range of the muscle fibers, okay, the diameter of muscle fibers, it ranges from, for young males, from 14 to like 46. Yeah, that's 46. Okay, that's 46. Okay, now for the adults, okay, if you check here, this is 20. That's from 22, right? Look at it. So looking at the range. So if this is 14, it's for young is 14. So looking at differences to 46, that's the range for the diameter of the young, 46. All right, because it's good to cite code data. The adult miles is from here, which is like, okay, 15 to about 80. Wow, that's much. Okay, so see the range, 15 to, is that 15? No, that's not 15, that's 20. From 20, yeah, 20 to 80. So which one has wider range? Okay, this is for adult mice. Adult mice has wider range of diameter. Okay, so the range of diameter is wider in adult, 20 to 80 is wider than 14 to 46. So that is one difference. Okay, another difference here is that if you check, look at it, so you see that most of these, all right, most of these young males, all right, you look at the middle, okay, you see that we have more of them. This is how many, that's number of fibers here is about, okay, about this is one of this is about 35. That's the highest number of fibers for young males. For adult males, the highest number is about this is 15. Okay, it's about 35, 15. Okay, so you see that that is the highest number of fibers. So if you look at this, you see that within this range is a smaller number because of the range. Okay, within falls within this range, so they are a bit similar. Okay, compared to this is a wider range, and they have more number of the muscle fiber, right, of young males. You can quote the data. Okay, so this is describe two differences between, we have described it. We looked at the range and we looked at the number, and then answer suggests how these differences may affect sliding filament model. Now, sliding filament model is a contraction of muscles, All right, you can see that for young mammals, all right, their diameter is small. See the range. Okay, so diameter of all right, of the muscle fiber is small. So if they have smaller diameter, I can see that the range is even, see, this is very small, 14. The highest is 46. So if they have smaller diameter, that means they will have fewer myofibrils, okay, or fewer myofibrils, because myofibrils make up the muscle diameter, muscle fiber. So smaller diameter means, okay, fewer myofibrils, and then fewer myofibrils means we're going to have fewer proteins, muscle proteins, like, you know, myosin, actin, and there's going to be smaller contraction compared to adults. Okay, so adults can run faster. Okay, remember we're connecting it to the topic. All right, smaller diameter means fewer myofibrils, okay, or sarcomeres. That means fewer proteins like myosin and actin. That means slower contraction, shorter contraction, okay, of the sarcomere. All right, so let's see what the mass scheme says. Right, so it says this is the first one, the difference. Young mice have more muscle fibers. Yeah, they have more muscle fibers that are similar in diameter. Yeah, 30. Young mice have smaller range. Okay, then you have to quote your data. Then they say suggest the effect. Let's see. So see what they, so young males 35, okay, at 30. So many of them, all right, 35 of them, their diameter is 30. Adults, you can see 16 of them, the diameter is 50. Okay, so see the spread, all right, 16 to 40, 14 to 80. Okay, is that okay? So that is it. So look at how it affects the sliding filament model. Okay, so young mice have fewer muscle fibers. They have shorter diameter, so they will have fewer muscle okay, fibers, not like the number, okay, of myofibrils, sarcomeres, because fiber determines the amount of myofibrils and sarcomeres. So less muscle protein, and there's a weak contraction for the young mouse. Okay, so let's go to the next question. The next question says Fig 8.1 is a diagram of chloroplast. So this is chloroplast, guys. I want you to pause the video. What is Caie? What is the name of Caie? Caie is stroma, light, site for lighting. Okay. What about this L? That's thylakoid, okay? So you can see that. Okay, what about M? M looks like the lumen of thylakoid, right? And this is a stack of thylakoid. N is a stack of thylakoid, which is granule. Okay, so using letter K to N, complete table 8.1. Each letter may be used once, more than once, or not at all. So let's complete the table quickly. The table says high concentration of protons. So which of them has high concentration of proton? Okay, of course, that's lumen. Remember that during photophosphorylation, remember that what happens is that electrons, when electrons enter, when they undergo excitation, the emission, okay, so the electrons enter into electron transport chain. When enter into electron transport chain, they move from one carrier to another. Okay, this is carrier 1, okay, carrier 2, and they're moving down energy level. This is electron carrier 3. Of course, the final electron acceptor here is NADP. Okay, let's assume this NADP. All right, so the energy released by this electron is used to pump proton. Okay, this electron moving from one carrier to another. Okay, electron, okay, moving from one carrier to another. So they move down energy level. This is high energy level, this is low energy level. So the energy release is used to pump protons. This is very important. So protons are pumped from stroma, if you remember. Now look at this, guys. This is the chloroplast, okay? Double membrane. Chloroplast is double membrane. That's chloroplast there. Okay. So this is thylakoid. So protons are pumped from the stroma into the lumen of thylakoid. Okay. So look at it. Okay. So first of all, protons are moved. Guys, look at this. Protons are moved from the stroma, pumped into thylakoid lumen, and then sets up proton gradient. So we have more hydrogen ions within the lumen compared to the stroma. So that sets up hydrogen or proton concentration gradient. So when this asks us where we have high concentration of protons, so we have high concentration of protons where the lumen, so the lumen is M, that's M, okay? So that's M. There is a location of photosynthetic pigment. Where is it found? You see it. So location of photosynthetic pigment. Location of photosynthetic pigment. Photosynthetic pigment is located where? Is located on the thylakoid membrane, which is L. Okay, because this is grana, that creates large surface area for it. So this L or N, that's the membrane that is here. Remember that the lumen is where there's accumulation. The membrane is where we have photosynthetic pigment embedded on the membrane. So either L, which is the membrane, or N is a stack of the membrane. Do you get? All right. So site for light independence, that's the stroma. This is stroma. Okay. Site for light dependent is the same. Okay. Thylakoid lumen, thylakoid membrane, L or N. Okay. So that means that we're going to have something like this, guys. We're going to have, watch this. So here is going to be L. Okay, that's M. Okay, M. And here is L or N. Okay, good. And here is just K. And here is what? L or N. That's site for light dependent. Any of this. Okay. So make sure you are solving this question with us, guys. Do not go and check the math scheme. Make sure you're solving the question. Okay. So look at this. So let's look at what the math scheme says. Yep. See? M, L. Okay. Any of them. So let's go to the next question quickly. We have taken time. Chlorophyll is the main photosynthetic pigment in plants. Describe the role of other photosynthetic pigments found in chloroplast, in plant chloroplast. Okay. So chlorophyll A is the main, so that's the primary pigment. Okay. So remember that we have the accessory pigments. The accessory pigments include, you know them. Pause the video and then try to solve this question. Describe the role. Describe what are the main, all right, features, characteristics of the role. So you mentioned them. You know, we have accessory pigments, like chlorophyll B is an accessory pigment. Chlorophyll B. Okay. Chlorophyll B. Chlorophyll B. Chlorophyll B is accessory pigment. Yeah. So chlorophyll B is accessory pigment. Don't forget that. And not just chlorophyll B, we have xanthophyll. Okay? You know xanthophyll? Okay, chlorophyll B, xanthophyll, carotenoids. Right? What do? They absorb wavelengths that chlorophyll A, which is the primary pigment. This primary pigment is a reaction center. This chlorophyll A. So the other pigments are accessory, so they absorb wavelengths that the primary pigment, which is chlorophyll A, does not absorb, and then they pass on the energy of that wavelength, to the primary pigment. Okay, look at it. So first of all, they form the antenna complex. This antenna complex. The antenna complex. Can you see the antenna complex? So antenna complex, this light harvesting clusters, we call it photosystem. That's photosystem here. So if you check the photosystem, from here to here are the accessory pigment. They form the antenna complex. These are the accessory pigment. So what they do is that they absorb energy. All right, that primary pigment. This is primary pigment. Chlorophyll A is a primary pigment. There's some wavelength this does not absorb, so they absorb wavelength that primary pigment does not absorb, and then pass the energy of that pigment to chlorophyll A. So they say describe the role. So the role is that they absorb wavelengths that primary pigment does not absorb, and then pass the energy of that wavelength. They transfer. Okay, so when they absorb, they transfer the energy, okay, to the primary pigment. Because primary pigment is the reaction center. That is where photo excitation occurs, photo emission occurs. All right, and then can give example of them. Okay, xanthophyll, chlorophyll B, chlorophyll, you know them, chlorophyll A. No, chlorophyll A is of course the primary pigment. Okay. Okay, xanthophyll, carotenoid. All right, look at them. Okay, can you see that? So any of these, so any four, chlorophyll B, or carotene, or xanthophyll, carotene, these are accessory pigments. So they act as accessory pigments, part of antenna complex, part of light harvesting cluster. Okay, this light harvesting system or cluster. So they absorb light, all right, photons, pass the energy of that photons. Okay, remember they absorb to the primary pigment to chlorophyll A. So what type of wavelength do they absorb? They absorb different wavelength of light. Okay, wavelength not absorbed by chlorophyll A. Okay, all right, let's go to the next question. It says the absorption of different wavelengths of light by chloroplast pigment can be represented by an absorption spectrum. This is an absorption spectrum, a graph that shows wavelength, graph of wavelength against absorbance. Okay, all right. Okay, that's absorbance against wavelength. Yeah. So fig 8.1 is an absorption spectrum of an extract of chloroplast pigment. An absorption spectrum of whole chloroplast. So we have this is whole chloroplast. So this whole chloroplast contains all the photosynthetic pigment. This contains some of them, maybe one. Okay. So if you are asked, guys, pause this video and check this out. If you are asked to describe the difference between this and this, the difference between this, but you know, if you're asked to describe it, okay, let's assume if you're asked to compare means show the difference and similarity. Is that they have the similarity, that they have the same trend. Okay, they absorb certain wavelength together. Okay, they absorb certain wavelength and they do not absorb certain wavelength. But if you're asked to describe the difference, look at the difference here. Okay, but remember, if you want to do description question, make sure you find the value of one box. Okay, from here to here is 50. So 10 boxes gives us 50. So one box is 5. From here to here is what? 5 for 1. So one box, 5 boxes gives us 1. Okay, one box will be 1 over 5, which is 0.2. All right, so you can see that certain wavelength there. Overall, if you want to describe the difference, whole chloroplast absorb more. It has higher absorbance overall, okay, compared to the pigment extract. Then you start citing, you have to quote data, guys. Choose one major feature. Okay, look at here, look at this. If you compare this, see the absorbance here, look at this now. If you go like this is the absorbance here is 0. Okay, this is 1.8. And which wavelength is that? Look at the wavelength. Remember that one box is 5. So this is the middle. This is 525. 525. The absorbance is quite low for pigment extract. But if you take it up, okay, compared to this about 7. So this absorb more of this than this. Okay. So I said 2 points general. Okay. This has more absorbance overall, higher absorbance compared to this. Okay, and then I'll choose a particular feature of this graph. Choose this and this. This has low. Okay, this particular wavelength. If you like, I mention other wavelengths and then mention their absorbance to show that whole chloroplast absorb has higher absorbance than pigment extract, guys. Okay, then I say that if they ask you to explain why is that? So whole chloroplast has more pigment, absorb more wavelength. Okay, whole chloroplast has granular, large surface area, so it absorbs more wavelength. Okay, compared to pigment extract. So we have. Alright, so look at it. I know they're going to go there. With reference to figure 8 point, describe the differences between the two spectra. Suggest explanation for the differences. So we have done this question. The difference, you know, one has higher, of course, whole chloroplast than pigment extract. Okay, mention a particular value. Okay, and then you can see the reason is because it has more more photosynthetic pigment, it has larger surface area, absorb more water. Look at it. This is the first one. Absorption higher for whole chloroplast throughout. Comparative. Okay. So this is comparative. Greatest. Okay, look at it here. So here you can mention any of these. For each of those wavelengths, mention the absorbance, compare two of them. And you see that they have the lowest absorbance. Okay, the highest difference is at 525, that place we mentioned. Okay. Now why does it have more? This is the reason. Because pigments arranged for better absorption in chloroplast are stacked. So they are stacked. So they provide more surface area. So chloroplasts contain more pigment, absorb more wavelength. All right, look at this. Dopamine is a neurotransmitter released in some synapses in the brain. The release and action of dopamine is similar to that of acetylcholine. So if you remember acetylcholine, all right, you can deal with this. This is a fig 9.1 is a diagram of a brain synapse where dopamine is a neurotransmitter. That's it. Okay, so this is a presynaptic neuron. That's the postsynaptic. Okay, because the presynaptic always contains vesicles carrying the neurotransmitter. Can you see it? Okay, and of course the post always contains the receptor and the sodium channel. Right. Okay, let's see what I wanted to do. So you get familiarized. It said describe how the release of dopamine from presynaptic neuron can lead to action potential in the postsynaptic neuron. Describe here sequence of events, guys. So if you remember acetylcholine, remember what I said. Memory is the key in biology. So acetylcholine. So all you just need to do is that look at, they say describe how the release. So once dopamine is released, just like ACH, the dopamine diffuses across the synaptic. This is the synaptic cleft, this gap here. It diffuses across the synaptic cleft and then binds to its receptor. Can you see this ligand gated binds to the receptor here? When it binds to the receptor, then sodium ion channel will open. The sodium ions diffuse inside, okay, and they cause depolarization. Then depolarization leads to action potential. Simple. That's it. Dopamine diffuses across synaptic cleft. That's it. Binds the receptor on postsynaptic. Sodium ion channel open, right? Influx of sodium ion to the synaptic. Depolarization, all right. And if that depolarization is higher than the threshold, is the minimum. Okay. If it's higher than the threshold, then action potential is initiated here. Okay, that's synapses. Okay, let's look at that again. In brain cells, the amino acid tyrosine is changed into dopa, which is then converted to dopamine. Name another compound in the body produced from dopa. Yeah, that's dopaquinone. Remember dopaquinone? Dopaquinone. Remember dopaquinone? Where we have tyrosine is Ty. Okay, converted to dopa, right? That is during synthesis of melanin. So dopa, right, is converted to dopaquinone. So that's another compound, dopaquinone, right? Yeah, dopaquinone. That's it. Or melanin, of course, this will be converted to melanin. All right, let's go to the next question. In some brain synapses, the neurotransmitter gamma aminobutyric acid GABA is released. This results in an influx of chloride ions into postsynaptic neuron. Suggest and explain. See, suggest and then give reason whether an action potential would be generated in the postsynaptic neuron if GABA is released into the brain synapse. Remember that chloride ion is a negative ion. So pause this video and try to solve it. Okay? So pause this video, try to solve it. So remember that chloride ion is negative. So if chloride ion diffuses into the postsynaptic neuron, if it diffuses, it will cause hyperpolarization. Okay, if it is sodium ion that is moving into, cause depolarization. But because sodium ion is negative, it will cause the postsynaptic neuron to become hyperpolarized, or to cause it to become negatively charged, right, or become polarized. And once it becomes polarized, of course, sodium ions are not diffusing inside. And because they're not diffusing inside, okay, action potential cannot be initiated, right? So action potential cannot be initiated. It is initiated when positive ion, sodium ion, moves inside. All right, you remember that graph? Okay, yeah, if you remember that graph, that diagram. So when positive ions diffusing, so you see the membrane becomes more positive, and this is where action potential is generated. But if negative ions are moving, the membrane potential will fall and becomes, okay, hyperpolarized. Yeah, so no action potential will be generated. Okay, look at it. Any three from the following: chlorine influx, okay, more negative, stays negative. So post-synaptic membrane becomes negative, hyperpolarization remains polarized, not enough sodium ion, so less likely to reach threshold. Yes, it doesn't reach threshold, so no depolarization of post-synaptic membrane. No depolarization occurs. No action potential. 3 marks. Okay, the mouse Elsis is a large member of the deer family. Is a large member of the deer family. Okay. All right. It lives in temperate forests in North America and North Europe, where snow is present. Snow is present there for large part of the year. I like snow. Fig 10.1 shows an adult male moose feeding in a lake. So look at the guy. This guy is feeding on the lake, enjoying himself. But you should be careful because lion or tiger might be somewhere trying to feed on it, make it lunch. Okay, so let's see. The moose feeds on a plant in the lake called water milfoil. So this is a plant, water milfoil, Myriophyllum aquaticum. The moose and the water milfoil belong to the domain Eukarya, which includes the kingdoms Animalia for this animal and the Plantae for that plant. Describe the main differences between kingdom Animalia and the kingdom Plantae. This is basic biology. Pause this video and list the differences. Okay. Pause it, the video and list. Okay, now which of them have cell wall? Okay, plant. This plant have cell wall. This doesn't have this. Okay, which of them is autotroph? Plant makes the food. This guy is heterotroph. Which of them, all right, have nervous system? Animal. This doesn't have. Okay, which of them have chloroplast? This has. This doesn't have. Okay, which of them have large central vacuole? Plants. These have small temporal vacuole. Which of them, okay, very importantly, of course, which of them has central this? Okay, so let's look at it. Yeah, good. Don't forget it. No cell wall, chloroplast, heterotroph. This stores glycogen, stores starch. Nervous move from place to another. Okay, not permanent. Yeah. So don't forget it, guys. 4 marks. The measurement of the surface temperature of land and ocean can be taken from locations around the world. The mean global surface temperature for land and ocean combined can be calculated for a mixed time period. Scientists calculated the mean global temperature for the 20th century. The mean global temperature for each decade, 10 years from 1880 to 2020, the mean temperature for each decade was compared to the mean for the 20th century. For each decade, the differences in temperature was calculated. See, these guys are doing research. The calculated differences are shown in figures. So look at calculated. Okay, so this is years. This is calculated temperature. Okay, what's the general trend? Okay, general trend is that it increases, right? It increases. However, it fluctuates too. Okay, with a spiral, but at the end of the day, it increases. Okay, calculate the rate of increase in temperature per decade between 1980 and 2020. Show your working, write your answer. So pause this video and try to do it. Rate of increase. First of all, find your increase. Okay, so but remember what I told you. You must know the value of one box from 1980. All right, of course, Okay, this one is already noted. Here, you see that 5 boxes represent 0.1. So one box will be what? 0.1 divided by 5, which is 0.02. So 1980, let's do it, guys. So if you check 1980 here, what is the 1980 is, okay? Where's 1980, guys? Yes, that's it. Okay, 1980 touches this graph here. Okay, so this is 0.2. How many boxes? 1, 2, 3, 4. About 4 boxes above 0.2. That will be 0.28. Yes, check it out. 0.28. Okay, 28. Why for 2020? Help me calculate it. That's the last one. That's 0.8 plus how many boxes? 3 boxes. That's 0.86. Okay, so that is 0.86. 0.86. Yeah. So what is the increase? First of all, increase. Right, increase is 0.8. all right, 86. Get your calculator. Minus 0.26. That's the increase, right? Increase in temperature. Okay, so punch the calculator. I have mine here. Come on. 0.86 minus 0.2628, sorry. Yeah, that, yeah, 0.86 minus 0.28. So that gives us what? 0.58. So that 0.58 is for, right, 4 decades, right? 0.58 for 4 decades, guys. 4 decades. So once they mention rate, so okay, how much is for 1, which is over 4? 0.58 divided by 4. Okay, that's about 0. The answer is 0.145. 145. Okay, yeah, you see, that's how they got it. 0.145. Okay, let's look at the last question, guys. Moose population have decreased in North America since 1980. Suggest and explain reasons for decrease in moose population. This is asking you for threat to biodiversity. What are the things that threaten biodiversity? Number one, of course, is climate change. Because we're talking about climate here. Climate change can affect it. They're not able to survive at extreme cold temperature. Okay. Number two is predation. Number three, of course, is habitat loss. Okay, maybe diseases for, okay, and of course competition. That's it, guys. That's it. Anytime they ask you what affects biodiversity, that's it. Climate, less food, snow and snow cover, so more predation, more hunting by human beings, increased competition, loss of habitat. That is it, guys. All right, thank you so much for staying through. All right, so we're going to post more videos, and I'm assured that the videos are going to get better. All right, this is schedule. Let's hear what you think. Which area do you want us to do videos for? Okay, and please help to share these videos. Bye.